Hello there, and thank you so much for tuning into our symposium. The NASA SEAS 2025 Human Research Fellowship is proud to present our new complement of experiments investigating the microbiome in space and its associated effects on the immune system.
Hi, my name is Mariangelys Alvarado, and I'm from Puerto Rico.
Hey, I'm Carmona-Trejo, and I'm from Waco, Texas.
Hello, I'm Ciara Crilow, and I am from Ohio.
Hi, my name is Joaquin Cooke, and I'm from Illinois.
Hi, my name is Michael Grove, and I'm from Brownsville, Texas.
Hi, my name is Madalynn Martinez, and I am from Alamo, Texas.
Hello, my name is Annabelle Staton, and I'm from Denton, Texas.
Hi, my name is David Weisglass, and I'm from Plano, Texas. This is our mission patch, and it represents lab rats, which is us. That stands for Lunar Applied Biological Research and Analysis of Test Subjects. At the top, there's a red cross representing the health and ethics aspect of human research. At the bottom, there's a double helix structure emanating out of the rat's tail, representing life sciences. You can see there's 8 stars representing each of our team members, plus 2 more representing our mentors, Mr. Petty and Becky. And of course, there's a rat representing us, the lab rats. Our project name is A.M.O.R.E, which stands for Astronaut Microbiome Omics Reactivation Exploration.
Here's a bit of an overview on our experiments. Prolonged spaceflight exposes astronauts to stress, microgravity, and radiation, shifting the balance of bacteria in the gut microbiome. These changes can weaken the immune system through metabolic pathways that are linked to the reactivation of latent viruses, like dormant chickenpox, in astronauts. Because maintaining a stable microbiome and strong immunity is critical for astronaut health in long missions, we chose the research question: how does prolonged spaceflight lead to shifts in microbial composition over time and associate with changes in the immune system through viral reactivation?
So the risk we'll be addressing in our experiment is the adverse health effects due to host microorganism interactions, and also the health effects due to autoimmune responses.
Here are some knowledge gaps that have been identified by the NASA roadmap. We believe these to be the most vital for the future of spaceflight. First of all, NASA outlines that we need to learn more about how isolation and weightlessness changes in spacecraft affects human microbiomes and microbial virulence. Additionally, NASA isn't very sure about how radiation may affect human health and wants to further test and validate new technologies to monitor astronauts in their space flights.
A.M.O.R.E is a string of experiments that we will be conducting here on Hera Analog and on the Artemis flight missions. Our first experiment will be a microbiome-focused experiment, which during the missions we will measure the composition of the microbiome during zero gravity. Our second is a virus-focused experiment, which will measure the growth of viruses and its effects on the human immune system in microgravity. And our last one will be the radiation-focused experiment, measuring the probability of radiation causing dysbiosis in the microbiome.
How we're actually going to carry out this experiment is we'll recruit around 4 to 6 participants, and we're going to separate them. Half will be on Earth and half will be in space. And we will replicate the exact conditions that the people in space and the people on Earth are experiencing. So that being diet, food, sleep, and EVAs. Everything will try to be the same in order to minimize other variables that could maybe affect our measurements. And how we're going to take these measurements is we're going to specifically count the taxas and the ratios between certain bacteria in the gut microbiome. What we'll do is before the mission starts, we'll collect pre-flight measurements as a baseline of comparison for the ones in space and on Earth. And throughout the mission, to collect further data points, we'll have time-based fecal sampling and other omics data to measure both groups throughout the duration of the mission.
The second string of experiments is testing the growth of viruses in space and on Earth and comparing the results, as well as the growth of the human system and later the combined growth of both the virus and the human system. When in microgravity, our human body has a habit of having a weakened immune system. And there are multiple known viruses that remain dormant in the body and can reactivate in space due to a weakened immune system. Now, there have been a total of around 212 astronauts who have reported a reactivated virus, but only 6 who have had mild symptoms. But with longer space exploration in the future, a weakening immune system could cause more diseases to wreak havoc on the human body. We will be comparing the results from prolonged periods of exposure due to microgravity, radiation, and a change of oxygen levels to the samples we have on Earth.
For the viral reactivation experiment, we divided them into 2 groups. one on astronauts, and one on petri dishes, also known as cell cultures. For the first group, we will have astronauts on an Artemis mission, assess immune response, gene expression changes, and infection resistance, collect blood, saliva, and skin swabs at key time points, and track signs of virus reactivation and immune changes. For the second group, we will grow viruses infected cells in elevated oxygen, one set on Earth and one aboard Artemis. Compare how microgravity affects virus behavior and growth.
Rather than rediscovering the fact that radiation mutates the microbiome, it's important to discover the probability of such mutations. Which taxa might be more prone to radiation-induced mutation, what functions they perform, and how their functions may be altered following radiation exposure. The space environment delivers more radiation than here on Earth, sometimes 40 to 80 times more per year. That means the odds of radiation interacting with microbial DNA or molecules with unpaired electrons goes up. Even with repair mechanisms, mutations are still likely. That could mean beneficial microbes lose function, or worse, they become pathogenic. Our hypothesis is that certain taxa of microbes may be more susceptible to radiation-induced mutation than others. While we know that radiation mutates the microbiome, we have an idea of which taxa may be more vulnerable to radiation-induced mutation and how this may affect the gut microbiome as a whole. In order to determine which taxa of microbes are present throughout the study, we will also require fecal samples from the astronauts to study the microbial ribosomal RNA. While this method can't tell us where exactly in the microbe mutation is occurring, it can help us identify which microbes decrease or proliferate with time. To measure microbial function in real time, we will be using ingestible nanosensor capsules containing molecules that will either bind to or react with metabolites, such as short-chain fatty acids. This then generates an electrical signal, allowing us to monitor microbial activity in real time rather than relying solely on genomic sequencing. These sensors mimic how astronauts might monitor gut health autonomously for future missions. The astronauts will all wear external radiation dosimeters to determine individual radiation exposure, allowing us to evaluate the impact of radiation with respect to time. By exposing microbes on Earth to simulated radiation, we can test how the same types react in different conditions. This will help us identify whether the symptoms can be attributed to radiation alone, or if microgravity and oxygen levels may be playing a role. The NASA Space Radiation Labs provide a controlled exposure to different types of radiation, including high-energy charged particles that mimic those of cosmic rays. Having that controlled environment is important to linking radiation to dysbiosis patterns. Here's a timeline for all of our fecal, blood, saliva, and skin samples. We will take samples at 5 key points aligned with the phases of the mission for consistency. First, we will take samples pre-flight as baseline to compare the changes in space. Then, to address the cumulative effects of spaceflight over longer periods of time, we will take samples in the early in-flight stage, the middle in-flight stage, and the late in-flight stage. Then, to see if the astronauts continue experiencing any additional symptoms after they return back to Earth, we will take post-flight samples after landing.
We are exploring 3 vital areas: microbial composition, viral reactivation, and radiation detection. Our data collection strategy is designed for precision and comprehensive insights. For experiment 1, tracking microbial composition, we use 16S rRNA sequencing to identify the bacteria and archaea present. To understand what these microbes are capable of, we had shotgun metagenomics, revealing their full genetic potential. In experiment 2, viral reactivation, we employ a suite of tools. ELISA detects viral components or immune responses, flow cytometry analyzes individual cells, RT-qPCR quantifies viral RNA, and plaqueasates definitively measure infectious viral particles. Crucially, 16S rRNA sequencing bridges these 2 experiments. It helps us understand how microbial communities influence viral reactivation, revealing the complex interplay between them. For experiment 3, nanosensors and radiation, nanosensors assess metabolite function, while Geiger counters offer a reliable means to detect radiation.
As previously mentioned, A.M.O.R.E compares microbial shifts in space with Earth-based controls, analyzes the impact of space radiation in the microbiome and its potential to amplify other stressors, and it investigates how these changes correlate with viral reactivation and immune function through tech like nanosensors.
The experiments we came up with in this presentation will be vital to the future of NASA and space travel. The results from each experiment provided valuable insights into how viral transmission occurs in microgravity, which can contribute to the development of improved sanitization. Additionally, specific findings can be used to build a predictive database helping identify the likelihood of viral reactivation and cataloging the effect of radiation on astronauts. These experiments are a key step towards safer space travel, giving us the data we need to protect astronaut health on future Artemis missions and beyond.
As technology continues to grow, A.M.O.R.E can too. For future missions, machine learning models can be used to automatically recognize patterns that may be too complex or subtle for human analysis, such as electrical signals or chemical outputs from nanosensors. This allows us to identify potential issues early and address them before they severely impact astronaut health. Tools like this may be crucial for future Artemis missions and long-duration spaceflight.
Now we would like to take some time to acknowledge some important people who have helped us along the way.
Thank you so much to our project mentors, Patrick Petty and Becky Fritchie.
And to our program managers, Selena Miller, Laura Tomlin, and Margaret Baggio.
We'd also like to thank the many professionals that have helped us along the way, such as Phil Spriello, Bradley Hoffman, Andre Stewart, and Tyson Brunstetter. And we'd like to dedicate this to John Charles, whose work has inspired us greatly.
We would also like to thank the University of Texas at Austin Center for Space Research for supporting NASA SEAS and all that it entails.
My time at SEAS was truly impactful. I enhanced my data analysis skills by working with real data from NASA's Life Sciences portal, tackling critical issues astronaut space. This experience made me feel like I was genuinely contributing to safer future spaceflight missions, tackling all the challenges along the way with my team.
What I have taken away from SEAS is that I got the chance to both talk and connect with my peers and my mentors. I have learned more about aerospace and my possible place here at UT Austin, and I have formed bonds that I will never forget throughout my life.
SEAS has helped me solidify what I was never sure of. Listening to so many passionate professionals has reinvigorated my love for science and established my career path towards STEM. I'm so incredibly grateful for all the people I've met at SEAS for giving me an experience I'm a member for the rest of my life.
My time at NASA SEAS was life-changing. As someone new to STEM, I arrived with a lot of uncertainty, but when I got here and had the opportunity to do research with a group of students just as passionate, I realized that anyone could find their place in STEM. I'll be leaving SEAS more passionate than ever with new friends and lasting connections. I learned how to work with real NASA data and grew as both a scientist and a teammate. This experience challenged me in the best way, and I loved working with my team. They made it truly unforgettable.
SEAS has been one of the best opportunities for me. When I first came here, I wasn't very sure about what I was going to do or how much I was going to like a 2-week program. But thanks to the independence that they've gave me, I've learned to love this team and love this program. It's one of the best programs I've ever had.
I came into this experience unsure of what to expect, but after spending valuable time at the Johnson Space Center and the Center for Space Research, I've developed a newfound appreciation in the field of aerospace, further pursuing my aspirations and my career goals.
I came into SEAS, first of all, not even thinking I would be at any type of event like this, but I was fortunate enough to learn that I should never be afraid of making truly meaningful connections with people I don't even know. And I'm also extremely fortunate to be able to look back on SEAS as a good memory and have a group of people that share a same passion as me and know I work together well with them. So I thank SEAS greatly for that opportunity.
Thank you for watching our presentation, and